Chromio. I'll be your guide to epistasis. Let's get going. To begin, epistasis is the interaction of genes at different loci, and there are 6 different types. Single dominant, single recessive, duplicate dominant, duplicate recessive, duplicate interaction, dominant recessive interaction. But today's focus is just duplicate recessive.
Duplicate recessive means that when the recessive allele is at either loci, it will mask the expression of the dominant allele, meaning that you can have a and little b resulting in the same phenotype no matter what the other locus has. This results in a 9:7 ratio.
That's quite a mouthful, but don't fret. I'm here to break it down for you. We'll use an example of flowers. A dihybrid cross, meaning 2 traits between a purple flower, big A, big B, and a white flower, little a, little b, produces an F1 generation of white flowers and purple flowers. If we cross the F1 generation with itself, big A, big B, and big A, big B, we should still have a ratio of 9 purple flowers to 7 white flowers.
So here's our first cross. We have big A, big B, crossed to little b, b. If you look at our table, we don't quite have a 9:7 ratio yet. That's because it comes in with the F1 generation. So here's our F1 cross. Big A, big B, crossed to itself. Big A, big B. Now you can tell that there is a 9:7 ratio. 9 purple flowers to 7 white flowers. Dominant to recessive.
Thanks for sticking with me. Hopefully you learned something fun about epistasis. See you next time!